So when we talk about cells, again, I'll have to write with my finger until I get the pencil recharged. Cells have membranes, and we talked about membranes a lot. We know that cell membranes define the cell, right? We know they're a phospholipid bilayer. These are things you should know. They should be already in your notes. I'm not writing them down. But do you know that they should be phospholipid bilayers? You know the different kinds of proteins that should be in there. And we've talked enough about that you should be aware that that's important. And we've said something about the idea of there being some kind of way of telling what gets into a cell and what doesn't get, and what gets out of a cell. We've also talked about this thing called, we mentioned, not talked about, we're going to talk about it today a little bit. We've mentioned this area, this space on the outside of the cell membrane. It's outside the cell membrane. It has a name. I'll give you the name in a second. And this stuff, it's made of proteins, and they're attached to the cell membrane. They can be tethered. The proteins can be anchored. There can be all kinds of ways they're attached. But there are these things called glyco, meaning sugar, right? Lipids. So maybe these are lipids that are tethered to sugars. There are these things called glycoproteins. Glycoproteins again are sugars that are attached to what?
Protein.
To proteins. And these both act as signals, almost like a key in a lock or a flag. So why would anybody put a flag out? Why would someone, there's a war going on, there's shooting back and forth. Why would you want to have a flag? To claim an area, I like that. What else? What's another reason you'd have a flag? To tell which territory is yours, I like that. So you're demarcating that something belongs to you, right? So another way these can be used is as flags. So again, what do we do with flags? We mark territories. We mark territories. This is ours, right? We mark what is ours. We mark what's ours with a flag. So when we talk about glycoproteins and glycolipids, they could be involved in signaling. In signaling what? In signaling what belongs to us. Someone close the door, please. What belongs to us? It could be a lock and key. It could be an enzyme in that space outside the cell wall, outside the cell membrane. What belongs to us? And one example of what belongs to us is this idea of blood groups. You've heard of blood groups before?
Yes.
What blood groups have you heard of? What blood groups have you heard of? For example, of what belongs to us. What blood groups? A, B, O, AB. You've heard of these, yes? And then there's plus or minus. It's RH, RH plus or RH minus. There's actually thousands more. There's thousands. I'm going to say thousands. It might not be thousands, but let's not say thousands, say many more. Many more markers, we call them markers, that tell us, they act like flags telling us this belongs to us. So if you're going to get some kind of transplant, you need to have some very similar markers. And so to get a transplant, they have to test all these different markers that are on the outside of all your cells. Because if they're different, your cell will say, this doesn't belong to us. We got to kill it. There's so many markers that no two people have the same markers. They don't exist unless they're twins. Unless you're twins, unless you have a clone. A twin is a clone. An identical twin is a clone. And the only way you're going to get the same markers on the outside of every cell is if you have a twin. So unless you have a twin, you have no donor that's perfect. So no matter what, we have to when you get a transplant, you have to take medicines that calm down your immune system. They kind of make your immune system drunk, if you will. So they can't see everything clearly. It's a little fuzzy. If we can do that, then we can transplant. But even then, you have to get it close. So we can't turn your immune system off completely, if that makes sense, right? Because then bacteria would kill you, correct? So viruses and bacteria are going to kill you if we turn off the immune system completely. So we got to do something about that, and the only way we can do anything about that is just to make your immune system act a little funny. Just calm it down, tune it down. So these markers, the way they work is with these glycolipids. Imagine taking a lipid. Remember, it's a carbon-hydrogen chain. The carbon being delineated by them, and then and at the end of that, you add a lipid, a carbohydrate. To that lipid, you add a carbohydrate. So you do something like this. Let me make it smaller. So you do something like this. You add a bunch of carbohydrates. They don't all have to be the same carbohydrate. Maybe the carbohydrates are in a ring, or maybe they're in a double ring, or maybe they're in a chain. There's all kinds of things that could happen to this. What's really cool about these glycolipids and glycoproteins is they can take on a bunch of more different shapes. And then each of these ends up being a different flag. That's how you can have 1 billion people, more than 7, 8, 9 billion people, and everybody have a different set of markers. Because each protein, each lipid that sits on the outside of that space can have a different shape. And that's because, you could add all kinds of carbohydrates and lipids on the outside. There's so many different combinations. This purple space on the outside here is known as the extracellular matrix. Why is it extracellular? Why extracellular? Why is it extra? Say what now? Very good. Because it's on the outside of the cell membrane. So the cell membrane's here. Everything inside the cell membrane is part of the cell. This is outside the cell, so it's extra. It's an extracellular matrix. It's attached to the membrane, but it's on the outside. And you'll see drawings with all kinds of crazy things in here. Think of it almost like a jungle out here of different molecules that stick out. Some are receptors, so they act as some act as enzymes. Some act, the lock and key idea that's happening in the extracellular matrix. Some act as signals. Some are being, like I said, receptors, or maybe they're markers, flags. But all that's happening on the outside of the cell. We haven't even gotten into the cell yet. Now, inside the cell, we're gonna divide this into two different kinds now. We got eukaryote. Let's talk about eukaryotes. And yes, eukaryotes are spelled with a K or a C. One's British and one's an English spelling, American English spelling. I don't care which you spell it, but it should be something like this, eukaryotes. Eukaryotes, all cells, and I should mention this, all cells have a cell membrane. All cells have the extracellular matrix. Should I draw that? Why not? I think I can handle that. All cells have this extracellular matrix that act as a flag of some kind. Hey, this is one of us. Some of them have like a mucus layer, but there's this outside that's... Now, eukaryotes have something special, and this is going to be key if you're going to understand, and you're going to have to be able to identify the difference. Eukaryotes have membranes inside the plasma membrane. They have membranes inside the plasma membrane. Does that make sense? They have membranes inside the cell. Does that make sense or not? Yes? All right, so they have spaces. They have compartments. Remember that from the video, compartmentalization. They have many compartments inside. How do we define a compartment? How do we define a space in a cell? We use what? Say what? Yeah, how do we make a room in a cell? What do we use? What do we use to separate the spaces to call it a room? To call it a room, you have to have something separating it from everything else, right? Like this is a room, and there are these things called room walls, right, to separate us from the rest of the building. If you're going to have a cell, you have to have something that's going to separate this big space from any other space that you create in here. What would you call that? What's the barrier? What is the barrier? What barriers do we use? You're right. You're absolutely right. But I'll give you a clue. I already wrote it down. Membranes. That's right. We use membranes. We use membranes to... I really can't stand the way my finger writes on this. Let's see if my pencil is... Is it charged enough for me to be able to... So, membranes. Can't you take a piece of paper and unwrap the whole thing and put it down and eat it? That way we don't have to listen to the crackling every 5 seconds. She's so nice. She's trying to be real quiet about it, but every time she crinkles something, he's like, crinkle, 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 crinkle. And then it's chocolate, so there's chocolate all over her fingers. It's all over her pencils. She's licking her pencil. It's just a mess. It's cute to watch, though. All right, so membranes on the inside of the plasma membrane. So what's key is that this eukaryote has many rooms. One of the rooms that make it very distinctive is this thing. And what do you think that is? That's right. That's called the nucleus. That's called the nucleus. It is made of a membrane, creating compartments. Inside this compartment, inside the nucleus, you have DNA. In eukaryotes, it's linear. It's linear DNA. It has 2 ends, just like we talked about. There's another room inside the nucleus. There's a whole other room here that's called the nucleolus. Very similar. So there's 2 rooms Now, I'm going to do one of these things if you don't mind. I hope you don't mind, but if you do, it's too bad. It's going to look something like this. Have you seen these things before? They do it in books a lot. Have you seen that in books or not? What am I doing? Why am I doing this? That's right. I love that. Zooming in is correct. Yeah. Brilliant children. That's right. Zooming in. And when we can zoom in, what we see if I zoomed in, and let's say I made the nucleus, I'm going to make it purple. All right. Actually, maybe I shouldn't because I'll make it yellow. So I'm going to make the membrane yellow. This inner membrane yellow. The white one is the plasma membrane or cell membrane, right? Everybody get that that's a cell membrane? Yes, you'll have to identify what each is and what each does and the difference between it and the others I'll show you, yes. If we look at this one, which is called the nuclear envelope or nuclear membrane, then what we'd see is this membrane. It'll be made of lipids. Is there someone knocking on our door?
Yes, sir.
Who's knocking on our door? What's going on? Is it not time to leave yet, is it? Oh, someone went to the restroom. There are holes in the membrane. There are these holes. These holes are called nuclear pores. They allow things in and out of the membrane. You know what? If you don't mind, I'm going to undo that, and I'm going to draw it over here. And nuclear pores they allow, bless you, stuff in and out of the nucleus. Now, DNA can't leave. So DNA cannot leave the nucleus. Is that clear? DNA cannot leave, but RNA can. That's going to be very important. This is one of those things that foreshadows. You've read books before, I'm assuming, and you know these things. This thing called foreshadowing, where they tell you something later in the story, it comes back, and that's the bad guy, or that's the good guy, or that's the thing that was a secret to everything. Well, this is one of those foreshadowing things. DNA can't leave the nucleus, but RNA can, and these pores let things in and out of the nucleus. There's another organelle in this eukaryote that's called, it looks like this. Let me see, color-coded. I'll make it blue. There's a lot of these. And again, they have a membrane. They're folded. There's two membranes. There's double membrane. So this one has a double membrane. And it's called a mitochondria. And mitochondria have double membranes, mitochondria process energy, they make ATP. We'll talk about what ATP is later. So you should know that's why they call it the powerhouse of the cell. You've heard of that before, I'm sure, in elementary school. They make process energy, the double membranes, and they have their own DNA. That's really interesting. They have their own DNA. So mitochondria are really, really interesting little things. And they're very much like prokaryotes. Are they prokaryotes? No. But are they like prokaryotes? Yes. And they're inside a eukaryote. Isn't that interesting? There are these things that are organelles that have double membranes, their own DNA, and they act like prokaryotes in very many ways, like very similar to prokaryotes that live inside every eukaryotic organism. Yes? Question?
Can they be mistaken as prokaryotes?
That's a good question. I suppose if they were floating around somewhere, somebody might consider them prokaryotes, but they're so distinctive. They can't exist outside the cell. So it's a good question. They're very similar to prokaryotes in many ways, these organelles. Now, that's one type of eukaryote. There's another, let's draw another type of eukaryote. Let me see if I can do this. I don't know if it's gonna, I'm gonna hate doing this, but I'm gonna go ahead and duplicate it. Now, there's another type of prokaryote that's very similar. Everything's the same. I'm gonna go ahead and get rid of a lot of this though so I can draw some of the other stuff. So a lot of this stuff is still there. Is that clear? I'm not getting rid of that. The cell membrane's still there. I'm actually going to go ahead and leave the lines. It is a eukaryote, though. It just has something else, some other things that the other eukaryote doesn't have. Yikes, it's too thick. Oh, I'm sorry, I know I'm picky, but it just looks stupid. I'll just rewrite the whole thing. How's that, jerk? It's still a eukaryote, but it has this really interesting set of features. And the first feature I'm going to go ahead and draw out is outside the cell matrix, it has this thing called, and it's not really to scale, so I apologize for that, but this thing called a cell wall. It's made of cellulose. So it's actually made of sugar, right? It's a polymer of sugar. We learned that. It's made of cellulose. And inside the cell, this eukaryote has something else that's weird. It has, let's see what color should I make this? These things called, I'm going to go ahead and they look weird. They have stacks of membranes. They're stacks of membranes. Again, double membrane. Let's go ahead and draw that out here. They have double membranes. They have their own DNA. They also process sugar and process energy. But they make sugar. They do something called photosynthesis. And they're called chloroplasts. And they're green. That's what makes plants green. And then there's another organelle that's really weird. That's not in the other eukaryote. It's really big. It's actually the biggest organelle in the cell. It takes up a good portion of the cell, of this type of cell. These are not in this, so let me erase that. I'm going to leave the chloroplast there because I already drew the line. So I'm going to erase these. These aren't inside. The chloroplast is not inside it. Is that clear? It's above it, if that makes any sense. Does that make sense? Yes? Are we clear on that?
Yes.
Yes? People in the back? Do you understand what I'm saying? So, no. They're still all inside the cell, but this is three-dimensional. So this is further back, right? So this organelle would be further back. This is closer to you, and this one's further away from you. Does that make sense? All right. I hope it does. I hope you don't confuse it. This thing holds a lot of stuff, and we'll talk about what it holds later. But it really has a lot to do with water. And this thing's called the central vacuole. Central vacuole. So as a central vacuole, has chloroplast, has a cell wall, but it still has a nucleus and all the rest of the organelles. There's a lot of organelles I still have to cover, but this is what we have so far. The central vacuole has to do with water regulation. That's the main function. It regulates osmosis, osmoregulation. And that's not a Q, that's a G. So that's water regulation. Are we clear? That's what the central vacuole is doing. So these two things, let's go ahead and look at them. They're both eukaryotes. This one, they both have mitochondria, correct? They both have cell membranes. And honestly, someone said in the other class they should do a Venn diagram. She was joking, but then it turned out I kind of agree with her. I think she said it because she was sick of Venn diagrams. But mitochondria, they both have nuclei, correct? They both have nucleolus. Can't you guys just leave the door open when you go to the bathroom? They have, huh? Yeah, leave it open. Yeah. Is it me or is it cold in here? It's either cold or hot, man. I can't get it to stay in just normal. So we know that these are all the things they share, correct? They both have an extracellular matrix. Again, they both share other organelles. We're not there yet. But then there's some stuff that's different. And now the stuff that's different, that's all the stuff that's the same. The stuff that's different is what? This one has, I should tell you. These cells, these types of cells over here, they can have a flagella, or they can have cilia. Cilia or flagella. Someone check the spelling of flagella. See if there's two L's in it. One of those things I always mess up. So flagella and cilia, they're for moving. Flagella you've seen on a sperm, right? They kind of have tails. They look like this. Well done. All right, good. And cilia, they're like little oars. In fact, I think Nolanix found ciliate the other day, was zooming around in the pond water. They zoom around, they eat bacteria. But you have cilia too. You have cilia in your throat. That's what feels like it's tickling. It's in your trachea and in your bronchioles. in your airways. You have these little cilia that help get the mucus out. When you cough up that mucus, that's the cilia moving all that mucus up with trapped dirt and bacteria out of there. So cilia and I should add that. So one of the things that's different is that, so these are the same, is that these guys have cilia and or flagella. All right. But this other kind has cell wall. This has chloroplast. This has central vacuole. What is this kind of thing here versus this kind of eukaryote? This eukaryote versus this eukaryote. Anybody know? No guesses? This would be known as a plant cell. And this one over here would be known as an animal cell. So one list of eukaryote, one type of eukaryote is an animal cell, another type of eukaryote. Both plants and animals have nuclei. They have these compartments inside the big compartment called the cell. So let's go ahead and talk about all the different kinds of eukaryotes and prokaryotes. Let's organize these into groups. So, because we're looking at a lot of cells, and Thursday you'll be dealing with a lot more cells. You'll actually have a formal lab write-up you're going to have to do on what you've discovered. So prokaryote can be either a bacteria or it could be an archaebacteria. Slightly different, but what's interesting about these guys is they have no internal membranes. They only have the plasma membrane, and they have round DNA, which is interesting to me. Their DNA is circular. They have smaller plasmid DNAs. Ribosomes, they have ribosomes, but ribosomes don't have membranes. So no internal membranes. So what does that mean? No compartments. No specialized compartments. Now in eukaryotes, we already talked about that. You have the nucleus, the nucleolus, you have the chloroplast and the mitochondria, and there's others we'll talk about a lot more. But here, there are no compartments, no membranes. Everything that happens in this one space. It's still a cell because it does have a cell membrane. And interestingly, it has a different kind of cell wall. It does have a cell wall, but it's different. All right, whatever. It does have a cell wall. It's just different from a eukaryotic cell wall. I shouldn't draw it that way. It's made of something different. A plant cell wall is made of cellulose. This is not made of cellulose. But it does have a cell wall. Has a cell wall, has a plasma membrane, has a nucleoid region. We call this region a nucleoid. Region, so there is DNA. It does have DNA. All living things have DNA. It has a plasma membrane. It is a cell, but they don't have all the fancy stuff that eukaryotes have. Are we clear? So the prokaryotes are very simple. And I believe your book calls the difference between this and a shack. A house and a shack, right? Or it's not the book, maybe it was the MIT lecture, but in any case. So if you think of a prokaryote, you can think of a prokaryote like a tent or a shack, right? One room shack. You got your bathroom, your kitchen, your bedroom, all in one space. There's no, your toilet's right next to the bed, it's right next to the stove, right? That's how people used to live. You realize that? Okay. Usually they put a toilet outside, but when they had indoor plumbing, the toilet would be right there. There's no room, no walls, nothing. It's just all in one space, as if we all lived in here. We had a kitchen there and a bathroom there, but there's no rooms. There's no separation. So but in a eukaryote, it's more like a modern house where you have rooms for everything, okay? Eukaryotes tend to have rooms for everything. And there's more than one type of eukaryote, just like there's more than one type of prokaryote. Eukaryotes, we know about animal, but we have animals. Animals have a really, really close cousin, which a lot of people don't think are close cousins. In fact, we used to think they were plants, but they're very close to us in how they do things. We call them fungi. So mushrooms are very close to humans. It's interesting. Their DNA, they have similar genes, they do similar things. They don't do photosynthesis. Obviously, they eat dead things, right? That's what mushrooms do. They're necrophores. They break down material, organic material. So they are obviously very different. But if you compare fungi, the mushroom to us, and then you compare plants to us, mushrooms are much closer. And then, of course, you have plants, and you have these things called protists. Now, that thing that you saw swimming around in the pond water on Friday, that was a protist. Protists are interesting. They're kind of in between a lot of these different groups, right? They can be a lot of different things. That you can have an amoeba that changes shape. It could look something like that. You can have a ciliate that looks like this. All right. You can have all kinds of different things. The key is that they all have what? They all have a nucleus. They all have organelles. They have all the other things that you'd expect to see in a eukaryote, but they're single cell. Protists tend to be single cell. Some are multicellular because they're so different. It's a big group, mostly. Thank you. Plants obviously are different from animals. I don't know why you put anything away. I'm not done yet. Let me finish my sentence. Rude. You should be writing it down. So, protists tend to be single cell. Plants obviously are a big group, and they do all kinds of things: trees, grasses, etc. Fungi, as you know, are like mushrooms, but they're also molds. You know, the stuff, that slime that grows in your bathroom is also a fungi. It just doesn't have a fruiting body. It doesn't produce a mushroom. That's all. And of course, animals come in all different sizes, including centipedes, by the way. Where's the what? What slimy stuff? If you've ever seen mold grow in a bathroom, you know, it's like a pink, or it could be All different kind. Yep. That you've never seen like a slimy coating in the bathroom at all. Any bathroom you've never been. Okay. Well, there can be slimy stuff in the bathroom. It's called mold. It's common.
I mean, I've seen mold before, not in bathrooms though.
Okay. Well, bathrooms are a perfect place for mold because it grow. It's very wet and often dark. So mushrooms need wet and dark.
I got an automatic light, so it's safe.
Well, whatever. The point is, mold grows in bathrooms and in basements and underground. All right, take it easy. Right.